Hello everyone. Today we'll be talking about the glycolytic pathway, the enzymes and reactions involved in this pathway, the energy production of this pathway, the inhibitors of this pathway, the importance of this pathway in the body. What is glycolysis? Glycolysis is the breakdown of glucose into pyruvate. It occurs in the cytosol. A unique thing about this pathway is that it can function both in the presence and absence of oxygen. In the presence of oxygen, the predominant glycolytic product in tissue is pyruvate, which then enters the mitochondria where it undergoes oxidative decarboxylation to form acetyl-CoA, which is then further oxidized in the citric acid cycle. In the absence of oxygen, the predominant glycolytic product is lactate, which helps to regenerate NAD+. Glycolysis consists of a 10-step reaction. The first reaction involves the phosphorylation of glucose to glucose 6-phosphate by hexokinase or glucokinase in the liver. This step commits glucose to the glycolytic pathway, ensuring that it is available for energy production. Here are things to know about this reaction. 1. It is irreversible, and this is because the reaction is highly exergonic and releases a large amount of free energy. 2. ATP is used for the phosphorylation of glucose. And 3. Magnesium ion is a crucial cofactor which is required for the activity of hexokinase. The second step involves the isomerization of glucose 6-phosphate to fructose 6-phosphate by glucose phosphate isomerase. The enzyme glucose phosphate isomerase is also known as phosphoexose isomerase. Here are things to know about this reaction. 1. The reaction is reversible. 2. It is an aldose-ketose isomerization since it involves the conversion of an aldose to a ketose. 3. No ATP is consumed or produced. The third reaction involves the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1. This step is known as the committed step, and the enzyme phosphofructokinase 1 is known as the rate-limiting enzyme. The enzyme phosphofructokinase 1 is allosterically inhibited by ATP, citrate, and other molecules, ensuring glycolysis is slowed down when energy level is high. Here are things to know about this reaction. 1. The reaction is irreversible. 2. 1 ATP is utilized for phosphorylation. 3. Magnesium ion is a crucial cofactor which is required for the activity of phosphofructokinase 1. The fourth step involves the splitting of the symmetrical fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyestone phosphate by the enzyme aldolase. Here are things to know about this reaction. 1. The reaction is reversible, and also in this reaction, no ATP is expended or produced. The fifth reaction involves the isomerization of dihydroxyestone phosphate to glyceraldehyde 3 phosphate by triose phosphate isomerase, yielding two glyceraldehyde 3 phosphate molecules to proceed in glycolysis. The sixth reaction involves the oxidation of glyceraldehyde 3 phosphate by glyceraldehyde 3 phosphate dehydrogenase. Each glyceraldehyde 3 phosphate is oxidized and a phosphate is added, producing 1,3-bisphosphoglycerate and NADH. That is, a total of 2 NADH is produced per molecule of glucose oxidized. The seventh step involves the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate by phosphoglycerate kinase. In this reaction, phosphate transfer from 1,3-bisphosphoglycerate to ADP to generate ATP. That is, a total of 2 ATP is formed per glucose molecule oxidized. The eighth step involves the conversion of 3-phosphoglycerate to 2-phosphoglycerate by phosphoglycerate mutase. In this reaction, the phosphate group is moved from carbon 3 to carbon 2, forming 2-phosphoglycerate. The ninth step involves the dehydration of 2-phosphoglycerate by enolase, forming the high-energy compound phosphoenol pyruvate. The tenth reaction involves the conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase. In this reaction, phosphate is transferred from phosphoenol pyruvate to ADP, forming pyruvate and ATP. That is, a total of 2 ATP is produced per molecule of glucose oxidized. This reaction is known as a substrate-level phosphorylation, and this is because ATP is formed at the substrate level. Also, the reaction in step 7, which involves the conversion of 1,3-bisphosphoglycerate to 3-bisphosphoglycerate, is also a substrate level phosphorylation because ATP is formed at the substrate level. Now let's talk about the energy production. In the reaction involving the phosphorylation of glucose, 1 ATP is utilized. Also, in the reaction involving the phosphorylation of fructose 1,6-bisphosphate, 1 ATP is also utilized. In the reaction involving the oxidation of glyceraldehyde 3 phosphate to 1,3-bisphosphoglycerate, 2 NADH is produced. And we know that 1 NADH is equivalent to 3 ATP. Therefore, 2 NADH will be equal to 6 ATP. So, the energy yield in the oxidation of glyceraldehyde 3 phosphate is 6 ATP. In the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, 2 ATP is produced. Also, in the conversion of phosphoenol pyruvate to pyruvate, 2 ATP is also produced. That is, at the end of glycolysis, a total of 10 ATP is generated. Subtracting the 2 ATP utilized during phosphorylation, we have a net gain of 8 ATP at the end of glycolysis. Okay, now let's talk about the inhibitors of glycolysis. We have iodoacetate. How does iodoacetate inhibit glycolysis? It does this by blocking glycerolaldehyde 3-phosphate dehydrogenase by binding cysteine, and this halts the conversion of glycerolaldehyde 3-phosphate to 1,3-bisphosphoglycerate. We also have fluoride. How does fluoride inhibit glycolysis? Fluoride inhibits glycolysis by inhibiting the enzyme enolase, which forms a fluorophosphate complex, therefore halting the conversion of 2-phosphoglycerate to phosphoenol pyruvate. We also have oximate. How does oximate inhibit glycolysis? Oximate inhibits glycolysis by inhibiting the enzyme lactate dehydrogenase, and this indirectly slows glycolysis. We also have arsenate. How does arsenate inhibit glycolysis? Arsenate inhibits glycolysis by mimicking phosphate, forming unstable 1-arseno-3-phosphoglycerates and also reducing ATP. So now let's look at the importance of glycolysis. Glycolysis produces ATP and NADH for energy, supplies metabolic intermediates for biosynthesis, supports anaerobic respiration, and serves as a universal regulated pathway for energy metabolism in all organisms. Okay, now let's summarize what I've done. Glycolysis is the breakdown of glucose into pyruvate. It involves a 10-step enzyme catalyzed reaction. It occurs in the cytosol. It results in a net gain of 8 ATP. It is inhibited by iodoacetate, fluoride, oximate, arsenate. It is important for energy production, supports anaerobic respiration, and supplies metabolic intermediates. So I've come to the end of this wonderful lesson. If you find this video interesting, do here to like, share, comment, and most importantly, subscribe for more updates like this.